Hello, my name is Unassumption and I'm here to talk to you about my final year university project in case anyone's interested. It aimed to identify bacteria that could tolerate the wastewater produced by a biomass plant on campus. Pollution, as we all know, is a major problem in the modern world. The emission of greenhouse gases such as carbon dioxide into the atmosphere has contributed to the ongoing problem of global climate change. But, if anything, polluted water is a bigger threat than air pollution. It is estimated by the World Health Organization that 14,000 people die every day from drinking polluted water. The economic costs to fisheries, tourism and other industries are very great, and the damage to aquatic ecosystems is often irreparable. Traditionally, we've used chemicals to address polluted water, but the chemicals we use to deal with pollution can themselves become pollutants, so more and more we're moving to bioremediation as a solution to pollution. Bioremediation is the use of organisms to degrade toxic pollutants, toxic chemicals, into more benign ones. In the example you see before you, the marine bacteria Alcanivorax is feeding on an oil droplet from an oil spill and making carbon dioxide. It's using the oil as a food source and producing carbon dioxide as a byproduct, as its product. The UEA has a biomass plant on campus which uses locally sourced wood chips as a fuel. It burns them at very high temperature, 1000 degrees on the diagram, and low oxygen conditions, which produces very little carbon dioxide, and also a biogas. The wood is basically vaporized into this carbon-rich gas, which is burnt in a power generator to power the campus and also to provide heat for the campus. This benefits us in three ways. The locally sourced wood chips decrease our reliance on fossil fuels, which are getting a bit scarce and pricey. It decreases our carbon dioxide emissions because there isn't much oxygen to react with the carbon in the wood and produce carbon dioxide. So this decreases our carbon dioxide emissions by 35%. And it also, because the power is generated on campus, the heat that's generated alongside the power can be used to heat the buildings on campus, making us more energy efficient. We don't need to burn as much fuel to heat ourselves. But there are downsides to this biomass plant. Water is needed to cool and clean the biomass generator, and this becomes polluted. Primarily, a large constituent of the pollution, 10% of the pollutants in this water, are phenol, which is a problem, not only because phenol is toxic to humans and many aquatic organisms, but because there are legal limits to how much phenol you can dispose of by pouring it down the drain. This is because phenol interferes, it kills bacteria, and bacteria are used in the sewage plants that deal with the municipal waste that's poured down the drains. Bacteria bioremediate our normal waste. They gobble it up and turn it into something less toxic, but phenol would kill them. The bacteria in our sewage plants are called activated sludge, and the phenol kills those things. So you can't legally pour concentrated phenol down the drains. There is some phenol in your soap, but that's like 0.4% phenol, whereas the stuff in the biomass plants wastewater is much more concentrated, and you can't pour that down the sink. So instead the university has to send it off to a specialist company that charges them a fairly hefty fee of £700 for every day to dispose of the amount of wastewater that's produced every day the plant is running. Costs the university £700. So the university has been working on finding other ways of disposing of this wastewater and of phenol that are less costly, and my project is part of this wider effort. The specific aims of my project are to identify the bacteria that are present in the wastewater from the biomass plant, and to identify bacteria that are capable of degrading that wastewater, especially the phenol in the wastewater. Before my project began, another researcher took wastewater and he incubated it in an aerated bioreactor at ambient temperatures for 4 weeks, and noted that the total carbon content of the water, the total amount of pollution in it, decreased by 50%, over those 4 weeks, and the amount of phenol in the water decreased by between 20 and 25%. He ran 2 reactors, and in one it decreased by 20% and in the other 25%. And this implies that something is degrading that wastewater. He also combined the wastewater from this bioreactor with a chemical that makes bacteria stained. It makes them purple, and as you can see in the picture on the right, that this confirmed that bacteria were present in the water in this bioreactor. So then I come into the picture now. I took some water from this, some of the wastewater that had been incubated in this bioreactor, allowing the bacteria to grow to a fairly high concentration in there, and I tried to extract the DNA from it so I could try and identify what the bacteria were by sequencing some of the DNA. And firstly, you need to get the DNA out of the bacteria, and so we tried 3 different methods I tried 3 methods to break open the cells and purify the DNA. I tried freezing them and heating them to crack the cells open. I tried using chemicals to degrade everything in the cells that wasn't DNA, and I tried basically shaking the cells with little glass beads to break the cells open, and then use a column which binds DNA and nothing else to separate the DNA from everything else in the liquid. Like the broken open cells are separated from the DNA in a column, and then you wash the DNA off this column that binds to DNA. But sadly, none of these methods work. There's a chemical called ethidium bromide which glows in the presence of DNA. It fluoresces in the presence of DNA, and we took this slab of jelly basically and put the DNA, and there were several like little wells dug in the end of the jelly. We poured part of our sample in those wells, the wells labelled on this slide with asterisks, and then run an electrical current through the gel which makes the DNA move through it. It moves slower if it's bigger bits of DNA, and in the lane labelled with a little lambda symbol, there are little bits, a mixture of DNA was put in that well, a mixture of DNAs of different sizes that were known sizes, and this can be used to identify what size the bit of DNA we've put in there is, but that doesn't really matter. This experiment was just to confirm that DNA was successfully extracted from the sample. If it glowed, there was DNA, and as you can see, there's no glowing in the lanes labelled with asterisks, so the DNA was not successfully extracted by any of the methods we used. We tried them several times. It didn't work. To confirm that the method we were using could actually extract DNA, we took some of that activated sludge, the stuff used in sewage plants to degrade most municipal water, which is rich in lots of bacteria. There's diverse bacteria in there, but it's quite dirty and stuff. So it's to test that this method can work on some pretty unclean samples that some methods don't work with, and we cracked open those cells successfully, and as you can see from the AS line, which as you've probably guessed by now, we put the extracted DNA from the activated sludge sample in that lane, and it's fluorescing. We got DNA from that. So the method worked, but we couldn't get DNA out from the wastewater from the biomass plant, probably because there weren't many cells and there were chemicals that were interfering with the efforts to purify the DNA in the column. So instead, we changed our goals a little bit to try to find bacteria that could tolerate the wastewater. So we put the activated sludge, which we knew we could get DNA out of, in the wastewater, and we incubated it for a while, so that if anything that could be killed off, for a few days we incubated it, so anything that would be killed off by the wastewater that was in that activated sludge would be killed off, and the stuff that wasn't going to be killed off, that could tolerate the wastewater, would survive, and only that stuff would survive. And then we put it on a plate that contained lots of sugars that the bacteria like to eat, and we got lots of colonies on this plate, as you can see, and we selected 5 of those colonies and used one of the kits that had successfully extracted activated sludge earlier, and took the chromosomal DNA, we took the DNA out of those 5 cells, and as you can see, they're labelled PC1, PC2 to PC5 here. We successfully extracted the DNA from them, and they're the right size. They moved through the gel to the right band on the little lambda ladder. They're the right size to be the piece of DNA we wanted to extract. Well, you can selectively amplify genes so that you get like thousands and thousands of copies of one particular bit of DNA using this method called PCR, and we extracted a gene called the 16S ribosomal RNA gene, which is present in every single bacteria from all of our 5 colonies, and we amplified that one gene, and as you can see, the amplification was successful. It's glowing on the jelly, and we then sent that off to be sequenced, that one gene. Why did we choose this one gene? Because it's present in every bacteria. Its sequence can be used to compare any bacteria and find out how related they are, and it can also be used to identify any bacteria. And there is a big database of the sequences of this gene called on the NCBI website, and you can just Google your sequence to find out what bacteria you've got, and we went and did that on this website called BLAST, and found out that all of our bacteria were bacillus. They were of the genus Bacillus, genera like a cat or a monkey or something, so they're closely related, but they were different species. Well, 4 of the 5 colonies were different species. 2 of them turned out to have identical sequences of this gene, and combined with their similar looks, the colonies looked a bit similar. They were both orange. It was discovered that they were actually the same species. And we then plotted a tree of how related they were using this other bacteria, Streptobacillus, as an outgroup. We know that that's not very related to any of these, and this just shows that like their evolutionary relationship and like what's similar to what. Bacillus are all gram-positive. That means they have like just one big cell membrane. They have one cell membrane and a wall around it, and they also form spores. So the question came to our mind that perhaps they were forming these very resilient spores, and those were surviving the wastewater, but they weren't actually gobbling anything in the wastewater up. They weren't growing in there. They were only growing when they were plated and presented with lots of nice food. So to test that, and to test our second goal, which, if you might remember, was to see if to find bacteria that could degrade the wastewater, we put them in a test tube, that contained water and the right level of salt for bacteria, and also, but the only food available to them was the wastewater. 10% of the tube we put all 5 colonies in tubes that 10% of it was wastewater in one of the tubes. One of the tubes with 25% of the tube is wastewater, the other 75% is water. And to test that they could grow, we gave them glucose, which is like every bacteria's favourite food. And on here, the blue columns show how much the bacteria grew when there was 10% wastewater. The red columns show how much they grew when there was 25% wastewater, and the green columns show how much they grew when there was 20% like sugar in there, their favourite food. As predicted, they grew best when there was their favourite food, but they grew very well when there was wastewater, so they could use that as their sole source of energy and their sole source of like building materials to build all the molecules in them, their source of carbon. If it was 25% wastewater, the bacteria didn't do very well. Although they can eat the wastewater, there's something in it that's toxic to them if it's a high concentration. So, as you see, the red columns are quite low, and in that second colony, it's pretty much killed them off. So, we tested with no food as well, and we tested some other bacteria on the hypothesis that just your average random bacteria we used, E. coli, wouldn't be able to eat the wastewater if that's the only food available to it, and it would die. So we know that it can degrade something in the wastewater, these bacillus. The E. coli didn't survive as predicted, and when there's no food for them, there's no carbon sources, they all died. As you see, PC1 and PC5, the 2 colonies that had the same sequence for their ribosomal RNA gene and looked the same, degrade pretty much the same, so they are the same species. And well, we wanted to test next what is it in the wastewater that these bacteria are gobbling up. Is it perhaps the phenol, the thing we really want to get rid of? So next we grew them in a tube with just the phenol at the concentration that's present in the wastewater from the biomass plant, and we saw that they couldn't actually use that as their only food. If that's the only food available to them, they couldn't survive. So although they could tolerate that level of phenol, if you gave them another food source, if you gave them some sugar as well, they couldn't actually use the phenol as their food source. They couldn't degrade it. They could live when phenol was around, but they couldn't eat it. So for future research, we can degrade something in the wastewater but not the phenol, and a topic for future research would be to find out what it is they can actually eat. One way of doing that would be to insert random mutations into the bacteria. There are several ways of doing that, and find out and just grow lots and lots of bacteria, each with a different mutation of these bacterial species we've isolated, these bacillus, and make lots of variation in the bacillus and grow them in the wastewater and find the mutations that stop it growing in the wastewater, and then we can identify what gene has been mutated and check on databases, well, what's this gene involved in degrading, and you can use that to figure out what the bacillus have been degrading that's in the wastewater. Alternatively, we could analyse the wastewater to find out what chemicals are present in it, using chromatography, for example. You know, when you put paper in ink water, the different coloured, like water with ink in it, and you put paper in it and the different colours will seep up the page. That's chromatography, and you can use more sophisticated forms of that to figure out what's in a mixture of chemicals. And we don't actually know exactly what's in this wastewater. We just know that phenol is very concentrated there, and we could find out what chemicals are what other chemicals are highly concentrated in this polluted wastewater, and test the bacillus against those to see what they degrade. Another topic for further research would be to find a way of actually getting the DNA out of the wastewater, so we can address our original goal of finding out what bacteria are present in this rather extreme environment. We could try putting some sugar in the wastewater to increase the number of cells there. We could try diluting it to dilute out the pollutant, whatever chemicals are interfering with our extraction of the DNA. And if we got the DNA, then we could make a gene library of this 16S gene. There's lots of steps involved, but all it does is take this, if you've started off with extracting the DNA, you've got just straight from the wastewater, you've got all the DNA from all the different bacterial species mixed together, and there's a bit of work involved in separating the DNA from different species out. But you can do that, and then figure out all the species present in the wastewater, and that's something else for further research. We could test to see if combinations of the bacteria we've isolated could degrade phenol. Perhaps one of them can half eat the phenol, and another one can eat the waste product the other bacteria has made. This is called co-metabolism, where usually there's 5 or 6 chemical reactions in taking one food source and turning it into carbon dioxide, something really benign, and maybe one bacteria can do half of those reactions and another bacteria can do the other half of the reactions, and you need both of them to completely break something down. So testing combinations of bacteria on phenol is a next step. We could also try to identify what the best conditions for breaking down the wastewater. We know it's breaking down something in the wastewater, and what conditions make it break that down quickest, what temperature and acidity are best for the bacteria we've isolated breaking down wastewater. These are topics for further research. Thanks for listening, and if you've got any